Good morning. My name is Dr. Ben Newman. I am a molecular biologist who studies coronaviruses, and I put this together for an internal meeting, and there was a nice response to it. What I tried to do here is cover all of the vaccines that are close to completion to sort of give a really nice, detailed vaccine update for where we are. And in I think previous Ask Dr. Ben episodes, we had covered the Moderna vaccine, which is the messenger RNA that makes the spike protein, and they put a whole lot into your arm, but it works pretty well. And we had also covered the Oxford adenovirus, the one that's being fronted by AstraZeneca. Over in the UK, and yeah, that's a thing called an adenovirus. That's our little hexagon with a gene inside for the spike gene. And so that's the little gray thing is the little genome, and the green thing is the little spike gene. And then it shows a spike diagram next to it so you can see what it's making. So in this video, let's cover the 5 other ones that have published phase 2 data. Phase 2 being the second of 3 steps, 3 trial steps, and then after that there will be a formal approval step, and at that point a vaccine is properly approved and can be used within certain guidelines, whatever they decide at the approval. Alright, so what are the 5 we're going to cover? Well, if you look at them, you notice the same pictures coming up over and over again, because they're really very similar vaccines. They're just barely different enough that you can say, okay, there's maybe, one of these is going to work a little better than the other. But they're all based mostly on the spike gene, except for the ones that have whole virus. And they're just putting in whole virus as a relatively easy way to produce lots of spike protein. So yeah, they're all real focused on the spike, and they're just delivering it by different methods, either as a messenger RNA, or as purified protein, or as a adenovirus that contains a gene to make the spike gene, make the spike protein, or as the full virus, which has actual spike proteins on it. So let's go look at these things. All right, so there's one slide for each of these. I pulled out what looked like their most promising result in terms of antibodies and their most promising result in terms of T cells. So antibodies are these little molecules that are going to float around in your bloodstream and are also going to be present on the inside of your lungs to try to catch the virus and stop it before it actually starts an infection. So they're very useful things to have around. And there are some that are better than others. The ones that you really want are called neutralizing antibodies. Those, instead of just sticking to the virus, will actually block a virus from coming into the cell if you have enough of them. We think based on the size of coronavirus, let's see, if I remember right, it's somewhere around 40 antibodies we think it would take to actually knock out one SARS coronavirus 2 virus. And then over on the right side, we're showing the T cells. So these are going to be white blood cells. And I think the theme that you'll get through all of these is that everybody is doing a fairly good job at checking for antibodies and doing fairly similar tests to check for neutralizing antibodies. There's only so many ways you can gimmick that test. When it comes to the rest of the white blood cells, some groups are doing a lot, some groups are doing almost nothing, some groups are doing things that kind of look like something and are really not very useful. So yeah, it's the takeaway, as somebody said at the meeting yesterday, is that it's very competitive out there, and it looks as though they are each trying not to tip their hand too far. So they want to show just the bare minimum to be able to get to the next step without giving away any secrets or any extra data or anything that might make it particularly easy to compare one vaccine to another and lead to a preference of one vaccine over another. So some of these are undoubtedly a little better than others, and I've tried to make a table at the end. And this is difficult because not everybody is doing exactly the same tests. So there are lots of little stars next to some of the values because there are some things that are not quite the same about them. But yeah, we'll have a look and see what you think. Okay, so first of all, we have the CanSino vaccine. And this is from China. Beijing Institute of Biology were one of the ones that helped to actually build this thing. And the publication is up there for the phase 2. That's right up at the top. Here, we'll underline it. There we go. Boop. And so, okay, over on the left side, you can see that they have kind of a weird way of showing how much antibody they make. So they're saying, okay, we've got this is the rough level of antibodies we have at the beginning, and then the dot here is for what happens after vaccination. And all they're talking about is the proportion of the people involved in their study who had more than a fourfold increase in their antibodies. And a fourfold increase is not much. That's like the barely detectable level. So it could be anything from amazing, wonderful antibodies to barely detectable antibodies. But yeah, they're all reflected in a not very helpful little red dot that's right there. And so this shows that after the first round, they had one little, they had about 50% that made some kind of response, small or large. And after the second round, about a month after the second round, they had more. Actually, no, that's, sorry, day 14, and that's day 28. There we go. And so they have most people that are making some kind of response. Not everybody, but most people. And so take from that what you will. Let's see, other interesting features. So instead of... So the best studies that are out there actually look at the different kinds of T cells. And so you've got 3 major kinds of helper T cells called T helper 1, 2, and 17. Yeah, there's a story, but it doesn't matter. And you've got cytotoxic T cells. So those 4 kinds. And the best studies will separate each one of those out and look at how many out of a population of that kind of cell alone are able to react to the coronavirus, go out and hunt it down, which is kind of their job. Then you got papers like this one, where they're looking at total white blood cells of any sort, most of which are probably going to be things like neutrophils. If you know immunology, there's just a lot of those around. And so of these things, neutrophils or macrophages or what have you, they're showing that there's a little population of them. There wasn't much of a population before, but after they vaccinate, there is a population that are able to respond in the most basic way to the coronavirus. So all you can say from this graph is they did something. Yeah. And something's better than nothing, right? But something may not be enough to protect you from actual infection or from symptoms. And so doing a test like this is almost the same as not doing a test at all. But at least they're doing a little bit. You'll see some of the other tests are actually worse than that, if you can believe it. Okay, next vaccine we got to talk about is the Sputnik 5, or maybe Sputnik V, probably Sputnik 5 though, Russian vaccine. And this is a combination of 2 other vaccines that, it's the adenovirus 26 vaccines. That's this AD26 up here. That's like the one that Johnson & Johnson are pushing forward in America. I think they had just entered phase 1 and 2 trials. And I believe China had also experimented with the adenovirus 26 at one point, and also adenovirus 5. And I think the idea here is that these are both viruses you may have had at some point in your life. And if you have antibodies against the little capsule, they're just using these adenoviruses as a delivery mechanism. But if your immune system shuts down, one of the delivery mechanisms, the hope is that the other one gets through. And there you can see nice rising antibody numbers. So they get up to reasonable amounts, not as high as you get with the Moderna vaccine. But let's emphasize this again. The Moderna vaccine puts in a heck of a lot of material into a person. And that's probably the reason for it having such a strong response, but also fairly strong side effects compared to some of these. All right. And on this one, they actually did, I think, the best job. So for all of this people beating up the Russian vaccine, they've done the best job at actually showing how it works and what it does. So they've got antibodies, they've got neutralizing antibodies, and they've got 2 of those cell types. So all of the helper T cells, they lumped all 3 of those together. And so they're looking at those, and they're showing that, yes, those guys can react to coronavirus. And they're looking at the killer T cells, and they're showing that, yes, those can also react to coronavirus. So we've actually built some specific immunity. And then this thing down here, the interferon, that's similar to the graph in the last slide, but this is showing the amount of increase over whatever the baseline was, and the last one was showing the actual values. So you could work it out. You could figure out how to convert between these two, but these two at the top, this one that says A over a B and C over a D, those are the ones that you really want, I think. All right, third vaccine. So this is a messenger RNA with modified nucleotides in the RNA. And it only encodes a little part of the spike rather than the whole spike. So just the receptor binding domain, that's the RBD up at the top. And this is, I don't know, this looks like the kid that showed up late to school and didn't have his homework done and is just frantically trying to get something together through homeroom. I think they were maybe a little bit behind and wanted to get caught up. And so they are publishing, this is the bare minimum. This is very bare bones. Most of these studies will have, I don't know, 5 or 6 figures in the text, and then a supplementary data section of 40, 50 pages with maybe 10 extra figures in there, and all the extra data that, that they collected. This is pretty much it for this paper. It's, real basic as they go. So what they're showing here is they do get some antibodies, not particularly high levels, but they're only putting in part of the spike. And so, yeah, all right, I guess that makes some sense that you would get, the reaction to that part of the spike if you put in roughly the same amount. And down here we're looking at the neutralizing antibodies, and so it takes a while for them to come up. And these are okay values, like 267 is about average for these vaccines we're talking about. But you'll routinely see neutralizing antibody levels of 1,000 or greater in people that have actually recovered from COVID-19. And so all of the neutralizing antibody data that we've seen from the vaccines so far is a little on the underwhelming side, I would have to say. And then look over here. This is what they do for the T cells in this one. And all they do is show you how many there are per volume of blood. And so what you can see is that as they put in more of the vaccine, the number of T cells goes down, which you would say, whoa, that's kind of looks like a bad thing, right? We don't want to annihilate our immune system. It's probably just reflecting that these T cells are undergoing some sort of selection and that some of them are now going to be positive. But as part of this process, we kind of weed through a lot of T cells that are not able to bind to SARS coronavirus, and so the numbers can go down and then will come back up, but you'll be filling in the random T cells that you had before with very specific T cells. This is the idea. So yeah, this graph tells you pretty much nothing. It says that, again, something might be occurring, but it's not even guaranteeing that it's something specifically related to SARS coronavirus 2 or COVID. So I don't know why they even did that, other than to say, hey, look, we did something with T cells here. And again, these are just my opinions, my reads on these, but I'll share them with you. And if you want to know more, the links are on top of all of these, and a real immunologist would be able to go through this and squeeze a lot more detail out and probably do a lot better job. So this is the overview, and if you're interested, hey, take the deep dive. Yeah, fine. Okay, next one is a whole virus vaccine. So this is from CoronaVac, also in China. And they're just growing up boatloads of SARS coronavirus 2 and then inactivating it, making sure that it's not infectious, and then injecting it. And they've done tests on mice and different animals, even primates with this. And the strategy seems to be pretty sound. When they put in a lot of the virus, they weren't getting very bad side effects that you could see in an animal. I mean, people can tell you, ow, it hurts in my arm. It's sometimes more difficult to tell that with an animal test, but, there's something there. They also point out that when they did their phase 1 study, this is just a phase 2 study, unlike most of the others, which are combined phase 1 and 2, because they're trying to catch up with the leaders, the market leaders. The first one, they purified the virus, and so these are little tiny purified viruses. You have to go to the paper and try and blow it up, but you can't get it a whole lot bigger or better than that, even in the paper. And what they say is that actually they lost a lot of the spikes, and this is true. When you purify coronaviruses, it's very difficult to keep all those spikes on there because they are spring loaded and they are ready to go off. And basically, any purification involves a certain amount of bumping and jostling and changing the amount of salt in a solution or the pH of a solution, even unintentionally. And so, yeah, any of that stuff will spring the spikes. And so they point out that in the next time, they actually got many more spikes on there. And so they go from about 20 spikes. Now, 20 spikes on a virus, that could sound okay. But a fully occupied SARS coronavirus should have around 80 spikes on it. And we know this from counting the spikes on coronaviruses. When you get three-dimensional images of them, you can actually do this. And so the first time they had about a quarter of their spikes. The second time they're up to 40 per virion. So they got about half the spikes that they should have. So it's better. It's definitely an improvement. And they say it makes for a much better vaccine. Fine. All right. Fifth vaccine. This is, again, one of these inactivated whole coronavirus vaccines. And the weirdest thing about this one is, look at this. So here they have the amount of antibody, and this is the neutralizing antibody, the one that actually shuts down the virus. And they're showing that before the first dose, these people don't really have any antibody, and that's great. And 14 days after the first dose, they should have some antibody because you vaccinated them, and that's enough time to start making antibodies. And these people all have no antibody. And then they go right before the second dose, so I think we're like a month after the first dose. And now there's just enough people in this category that they have some antibody, but none here in the other doses. And so you're saying, wow, this is a vaccine that actually does nothing. Wow. But they give it to him a second time, and that gets him up to a low but reasonable level, around 100 neutralization titer. And then they do it a third time, so 3 vaccinations. And they get up around 250, 300, that sort of area in neutralization dose. So they got there in the end. Maybe that's the maybe that's the takeaway. But boy, did it take a lot of doses. And vaccine compliance with this one may be a problem if you have to. I mean, even in childhood vaccines, when you have to get parents to come in week after week after week to get their kid, the next vaccine every 2 weeks or every month, you lose some people. So once again, on this one, they didn't actually do the proper studies. Now, they did a super detailed version of studies that don't really help us much. And what I mean by that is they just counted the total number of each of these cells that is able to produce certain factors that's, got these things in it. So these IL-2 guys here, they're going to be like the Th1 cells. And you can see their numbers go down with vaccination, which could mean that we're selecting for some good ones. Yeah. Or, it could mean a lot of stuff. But they would need to do the test where they actually look to see if they made any specific ones. And they did not do that. Interleukin 4 is something that's going to be made by these Th2 cells. And again, they go down, so something's happening. And then interleukin 17 is going to be made by these Th17 cells. And again, they go down a little bit, not quite as much. So probably there's a bit of something happening here. Not as much as is happening with either of these. But are we actually making any T cells that can recognize the virus? We don't know because they didn't check. All right, so here we go. This is the table that was promised. And I've done my best to try to get all of these different studies roughly into line. So we've got the name of the thing, and this is the one from the Beijing Institute of Biology that I think is also being tested in conjunction with Wuhan Institute of Virology. I think I have that right, but we'll have to check the paper to make sure. This is the actual what's in the vaccine, and this is how many times they're giving it, and I'm picking out the high dose here. For some of these things, I'm trying to pick out, I think, generally the highest dose. This one, I think they went up to a 100 microgram dose, but I think their best result might have been at 30 micrograms. We'd have to go back and check that. And then I pulled out, okay, so this is the phase that they are in right now but are not completed with. And here we're looking at total spike antibodies at 14 days or 28 days after the last infection, or 14 days after the first infection and 28 days after the last infection, something like that, because there's different numbers of infections. But it's like the midpoint and the like early response and the ultimate best response, best you're going to do with these things. And so, yeah, there are some of these things that, produce pretty good numbers. So these are the messenger RNA, both of those messenger RNAs. And this one is one of the adenovirus vaccines where they give a quite high dose. And yeah, that seems to give pretty good numbers. Now, the Sputnik 5 vaccine got low numbers here, and they're actually giving the same dose, 10 to the 11th virus-like particles there, but they're only counting anti-receptor binding domain antibodies. And so a 5,000 receptor binding domain thing might mean a 50,000 total antibody thing, rough equivalent, but it is really hard to say. And there's no good way to compare these from the papers that we've got. Neutralizing antibody, I think, is the most comparable, and I picked out the best number that they were able to achieve with each of these, with whatever they said was the best vaccination. And you can see they approach 300, but don't quite get there. Whereas natural infection can produce, 1,000 and up of neutralizing antibody. And then over here we got our different T cells, and the T cells are just a mess, but the most complete and detailed ones are from Moderna. They did a nice job going through those, and from Russia, the Sputnik vaccine. They also did a real nice job, and I would say maybe even a peg above where the Moderna testing was. So, in terms of side effects, that's another thing that people would want to know about. If it's class 1 or class 2 side effects, these are just barely noticeable and mild. So these are not things that are worrying. And these are things that you generally have to ask a person. They're not going to self-report. When we get to category 3, this is now severe. This is the point where they would usually report this to you, saying like, wow, I had such a bad headache I couldn't get up for 2 days or something like that. But they didn't have to go to hospital. If they get to category 4, that usually requires hospitalization. So that is not just severe, but serious. And so... those are the ones you want to watch for. The Oxford adenovirus, I believe they had one person. So they paused the study. So from that, I'm inferring that they probably had one person who showed up with a category 4. And then they decided that it was due to something else, or it was, an underlying thing that they hadn't caught. And so they've restarted now. But mostly, the adenovirus vaccines tend to be class 1 and 2, except the one where they put in a ton of it and got a really good response. And so, you get a lot of class 3 side effects there. The mRNA vaccines, generally you get better numbers in terms of antibodies, but you get much higher side effects there. They're kind of a long list, and I've abbreviated these lists to fit in a little tiny box, but there are a lot more side effects there. And then the actual inactivated viruses are actually fairly benign. Just class 1 or 2 only, and this one paper, I dug through every last bit of the supplementary information, and I still couldn't figure out exactly how many class 3 events there were. They just said there were few of them. So few. Yeah, so take that with whatever grain of salt you want to take it with. So we've got a range of things. We've got a couple of messenger RNA vaccines that hit you hard, make a strong response, and have higher chance of severe side effects. And we've got in the middle, maybe the baby bear version. We've got the just about right adenovirus vaccines. We've got a nice range of those. And at the possibly too cold version, or maybe also right there in the middle, hard to say, baby bear, mama bear, I don't know. We've got the inactivated vaccines. So those are roughly equivalent, or maybe a peg under some of the adenovirus vaccines. All right, so there's your little explainer in graphic form. These are each of the things. This is what they seem to be best at. So antibodies, helper T cells, Th1 type helper T cells, and killer T cells, or these CD8s. We would assume there's probably going to be similar things for the T cells on this second vaccine, but they didn't do the tests. So yeah, you don't do your homework, you don't get the credit, right? Over here, we've got all of the different adenovirus vaccines. And some of these do a real nice job with T cells, such as the Russian vaccine. The others, they maybe haven't looked as much, but where they have looked, they find a pretty strong one-sided T cell response. So a pretty strong, what's called a Th1 response, which if you had to pick one of the helper T cell responses, that's probably the one you'd pick. So that's not such a bad one. Yeah. Sanofi and Johnson have both started and kicked up a lot of dust, but they haven't published anything yet in terms of clinical trials. And these are the inactivated vaccines. And we know they make antibodies, but until they actually do the tests and show us that they do something to T cells, they're probably doing wonderful things to the T cells, but we won't know until they actually publish some of the data. So there you are. That's kind of a rundown on where everything is right now. And so all of these in red or blue are in phase 3 at the moment. And so you'd expect phase 3 to take maybe, I don't know, 2 months, 3 months, something like that. It's going to depend how long it takes to get enough people that either get sick in the placebo group or don't get sick, in the treated group. And yeah, to where you have a statistically significant difference between the two. And that could be a while. And so I think that's going to be the slow part. Just giving the vaccines, some of these vaccines, you take a course of maybe 3 vaccines over a period of 2 months. So that's kind of the bare minimum amount of time that one of these tests could take. And then you want to leave a little bit of time after the second vaccine for, to see whether it actually worked, whether it prevented SARS-CoV-2 infection, or whether it prevented disease but led to infection. That's another possibility. So we will see, but there. Hopefully this is useful, and yeah, by all means, go in if you're really interested in the details and have a look at some of those papers that are linked in here. You can probably squeeze out a little more detail and more information and a little bit better information if you have some background in this area. That's all for today. Thanks very much. This has been Ask Dr. Ben.